Continue talking about the cell with talking about how the cell grows and reproduces. So section 10 talks 10-1 talks about how cells grow. Living things grow by producing more cells. And there are two reasons why cells divide rather than to continue to grow indefinitely. And the word indefinitely means forever. The larger a cell becomes, the more demands the cell places on its DNA. And DNA is deoxyribonucleic acid, which you learned about in chapter 2. The cell has more trouble moving enough materials and wastes across the cell membrane if the cell continues to grow indefinitely also. DNA overload is where there is not enough DNA to help the cell function properly. The information that controls a cell's function is stored in one molecule of DNA, and DNA is found in the nucleus. And we learned back in chapter 7 that the nucleus is the control center of the cell. When the cell is small, the information stored in DNA is able to meet all of the cell's needs, so the cell functions properly. As a cell increases, it does not make extra copies of DNA in order to help the cell function. If a cell grows without limit, an information crisis will occur, causing the cell possibly to stop working or shut down because it just can't help the cell function due to its size. The second reason was having trouble moving things across the cell membrane. Food, oxygen, and water enter a cell through its cell membrane, and wastes leave the same way. So we learned about diffusion, osmosis, and active transport as ways of getting things across the cell and out of the cell. If the cell gets too large, it's more difficult to get these sufficient amounts of oxygen and nutrients in and waste products out. So then, once again, the cell cannot function properly. So what the cell has to do is the cell has to divide. A growing cell divides, forming two daughter cells. And this is very important to remember. They are called daughter cells. So the two new cells are called daughter cells. Cell division is the process by which a cell divides, once again, into two new daughter cells. Before division occurs, the cell replicates or copies its DNA. And this is key, this point here is key to everything. You have to remember that before division occurs, the cell is going to copy or make an additional copy of its DNA so that the new cell has the same information as the original cell. Each daughter cell receives one complete set of genetic information. So they're going to be identical to one another. So how does this actually occur? Every cell, once again I'm going to stress how important this is going to be. Every cell must first copy its genetic information before cell division begins. This is extremely important to remember. So before division begins, the cell has to copy its genetic information. Cell division in eukaryotes is complex and occurs in two stages. The first stage is called mitosis, which is the division of the cell's nucleus. The second stage is called cytokinesis, which is the division of the cytoplasm. So once again, in eukaryotic cells, there are two stages. The first stage is mitosis, and the second stage is cytokinesis. Reproduction by mitosis is classified as asexual. And once again, reproduction by mitosis is classified as asexual since the cells produced are genetically identical to the parent. These are key things. In mitosis, it's asexual reproduction, and the cells that are produced are identical to the parent cells. Mitosis is also the source of new cells when a multicellular organism grows and develops. So examples here are going to be making new skin cells. For example, you burn your finger and your body has to repair that. So what your body's going to do is it's going to make brand new cells exactly identical to the cells that were damaged or destroyed. So examples are skin cells, muscle cells. These would be examples of when we grow and develop or something is actually damaged. That's how those particular cells are going to reproduce. Chromosomes carry the genetic information that is passed on from one generation of cells to the next. And chromosomes are made up of both DNA and proteins. And we learned about proteins once again back in chapter 2 also. And we said that the monomers to put proteins together were amino acids, and the monomers to put DNA together are nucleotides. Every organism has a specific number of chromosomes. For example, fruit flies have a total of 8 chromosomes. Humans have a total of 46 chromosomes. Carrots have a total of 18 chromosomes. Chromosomes are not visible in most cells except during cell division. So that's the only time that you're going to see these individual chromosomes. Well before cell division, each chromosome is copied. And once again, I think I've stressed this particular situation here. Well before cell division, each chromosome is copied. Very, very important. Each chromosome then consists of 2 identical sister chromatids. So when we're looking at the chromosome, this is one chromatid, and this is the second chromatid. So there's one chromatid here and the other chromatid here. And both of these are going to be identical. This is after the copying has taken place. So each chromosome then consists of 2 identical sister chromatids. Each pair of chromatids is attached at the centromere. So this central part here, right in the middle, that is attaching the 2 sister chromatids together. These 2 are identical to one another. All right, the cell cycle are the stages that the cell goes through when it makes a copy of itself. And what we're going to talk about in this half of the notes is we're going to just talk about the beginning portions of the cell cycle. Interphase is the in-between period of growth that occurs when cells are not dividing. And so hence, the word inter means in between. So that's what your cells are doing the majority of the time. The majority of your time, the cells in your body are in the interphase stage. The cell cycle is a series of events that the cells go through as they grow and divide. And the cell cycle consists of 4 phases. The M phase is for mitosis and cytokinesis. The S phase is where chromosomes replicate or synthesis of DNA. So S would be for synthesis, and synthesize or synthesis means to make DNA. G1 is cell growth. G2 is preparation for mitosis. So these are the 4 phases of the cell cycle: M, S, G1, and G2. Interphase can be quite long, whereas cell division takes place extremely quickly. An interphase is divided into 3 phases: the G1, S, G2 phases. Once again, during G1, cells increase in size and synthesize new proteins or DNA. So G1 is cells increase in size and synthesize new proteins. During the S phase, chromosomes are replicated and DNA is synthesized. During the S phase, chromosomes are replicated and DNA is synthesized. And during the G2 phase, which is shortest, organelles and proteins required for cell division are made. So, during G1, they increase in size, growth. During the S phase, chromosomes are replicating and DNA is synthesized. During G2, all of the rest of the organelles and proteins required for cell division are made. So once again, these are the 3 phases of interphase.